Hello, ladies and gentlemen. On behalf of International Online Conferences on Engineering and Natural Sciences, allow me to extend a warm welcome to you. Let me briefly introduce myself. My name is Hira Civelek and I'm 19 years old, and I'm delighted to be here today to talk to you about molecular structure and docking analysis of the integrin inhibitor cyclopeptide. I will start with introducing my team. We are 5 people in this team. Me, Hira Civelek, Mechatronics Department, Vocational School at Yeditepe University in Istanbul, Turkey. Sefa Çelik at Istanbul University, Faculty of Science, Department of Physics in Vezneciler, Istanbul. Demet Demirağ, Institute of Graduate Studies in Sciences at Istanbul University in Istanbul. Ayşen Özel at Istanbul University, Faculty of Science, Department of Physics in Vezneciler, Istanbul. Sevim Akyüz at Istanbul Kültür University, Faculty of Science and Letters, Department of Physics in Bakırköy, Istanbul. In this article's abstract part, all potential conformations of Cyclo with anti-cancer activity were found, and the most stable conformer was revealed using Spartan 06 program, utilizing the MMFF approach. And then the energy differences between the conformers were calculated, and the low energy conformers, which are essential to determining biological activity, were identified. And then, in this article's introduction part, integrins are transmembrane receptors that connect the actin cytoskeleton to the extracellular matrix, which is called ECM. Integrins are multifunctional heterodimers that regulate cell-to-cell and cell-to-matrix interactions via signaling and are involved in a number of critical functions, including cell cycle and proliferation control, in part through collaboration with growth factor receptors. As a result of mechanical chemokine growth factor signals, this link is dynamically reorganized. The V3 integrin acts as a receptor for vitronectin, a cell adhesive protein similar to fibronectin. As you can see here, the integrins and inhibitors relationship is like lock and the key. And this article's methods and calculations part, the Spartan 06 program and the MMFF approach were used to carry out conformational analysis. The CAVA program was used to determine the likely binding locations on the receptor's surface. On the discovered active sites, molecular docking investigations were performed using Autodoc Vena software. And here, obtained 5 lowest energy conformers of Cyclo by conformational analysis. And then the energies of the most stable 5 conformations of cyclopeptides, conformers, total energies, and the relative energies. Here, the first and total energy and the relative energy. The 3D doctrines of the most stable conformer of Cyclo in active site alpha V beta 3 integrin, and then alpha V beta 3 integrin is minus 10.5 kilocalories divided mole. And last part, conclusion, the most stable conformer of cyclic pentapeptide, Cyclo, was determined due to the importance of function-activity relationship for the bioactive molecules. The interaction of the tidal molecule with alpha-V-beta-3 integrin was investigated by docking simulations by using the most stable conformer of the Cyclo to assess its biological activity and examine the inhibitory effect on integrin. The binding affinity of Cyclo to alpha-V-beta-3 integrin was found to be minus 10.5 kilocalorie divided mole. According to molecular docking predictions, Cyclo found to have a good inhibitory effect on alpha-V-beta-3 integrin and may exhibit significant enantiomer properties. Thank you for your attention.